friends, I'm Anika Sabu, first year MSc Biology, St. Mary's College, Thrissur. My topic is ultrastructure of bacteria. Structure of prokaryote cell. Small size 0.5 to 2 μm. Large surface area to volume ratio. A variety of shapes. Outer cell wall very thick, made of specialized molecules. Cell membrane may have different consistency of molecular from eukaryotic cells. Ribosomes smaller, no organelles, no nuclear membrane. One double stranded circular loop of DNA. Prokaryotic cells almost always are bounded by chemical complex cell wall. Inside the cell wall and separated from it by a periplasmic space lies the plasma membrane. The genetic material is localized in a discrete region. The nucleoid is not separated from surrounding cytoplasm by membranes. Ribosomes and larger masses called inclusion bodies are scattered about in the cytoplasmic matrix. Both gram-positive and gram-negative cells can use flagella for locomotion. In addition, may have cells surrounded by a capsule or slime layer external to the cell wall. Functions of prokaryotic structure. Plasma membrane, selectively permeable barrier, mechanical boundary of cell, nutrient and waste transport, location of many metabolic process, respiration, photosynthesis, detection of environmental cues for chemotaxis. Gas vacuole, buoyancy for floating in aqua environments, ribosomes, protein synthesis, inclusion bodies, protein synthesis, storage of carbon, phosphate and other. Nucleoid, periplasmic substances, localization of genetic material, periplasmic space containing hydrolytic enzyme and binding cell wall proteins for nutrient processing uptake. Cell wall gives bacteria shape and protection from lysis in dilute solution. Capsules and slime layers, resistance to phagocytosis, adherence to surface, fimbriae and pili, attachment to surface, bacterial mating, flagella movement, endospore, survival under harsh environmental condition. Ultrastructure of typical bacteria. Cell wall. Cell wall is the layer, usually fairly rigid, that lies just outside the plasma membrane, give them shape and protect them from osmotic lysis. The cell wall of many pathogens have components that contribute to their pathogenicity. The wall can protect cell from toxic substance and is the site of action and several antibiotics. Peptidoglycan, the most important molecule in the cell wall of bacteria. The gram-positive cell wall consists of single 20 to 80 nanometer thick homogeneous peptidoglycan or murin layer lying outside the plasma membrane. The gram-negative cell wall is Q complex. It has a 2 to 7 nanometer peptidoglycan layer surrounded by a 7 to nanometer thick outer membrane. Peptidoglycan or murin is an enormous polymer composed of many identical subunits. The polymer containing two sugar derivatives, N-acetyl glucosamine and N-acetyl muramic acid, and several different amino acids, three of which D-glutamic acid, D-alanine and mesodiamino pimelic acid. Gram-positive bacterial cell wall and gram-negative bacterial cell wall. Gram-positive cell wall. Homogeneous cell wall of gram-positive bacteria is composed of primarily of peptidoglycan, which often containing a peptide interbridge. The teichoic acid are connected to either the peptidoglycan by a covalent bond with N-acetyl muramic acid or to plasma membrane lipid are called lipoteichoic acid. Gram-negative cell wall. The outer membrane lies just outside. Outer membrane lies outside the thin peptidoglycan layer. The most abundant cell membrane is butyl lipoprotein, covalently joined to underlying peptidoglycan and embedded in outer membrane by its hydrophobic end. Constituents of the outer membrane are its lipopolysaccharides. Outer membrane is more permeable than plasma membrane due to the presence of special or in proteins. Capsules, slime layers, and S layers. Some bacteria have a layer of material lying outside the cell wall. When the layer is well organized and not easily washed off, it is called capsule. A slime layer is one of diffuse, unorganized material that is removed easily. Many gram-positive and gram-negative bacteria have a regular structural layer called a S layer on the surface. The S layer has a pattern something like floor tiles and is composed of protein or glycoprotein. In gram-negative bacteria, the S layer adheres directly to the outside membrane. It is associated with the peptidoglycan surface in gram-positive bacteria. It may protect the cell against ion and pH fluctuations, osmotic stress enzymes, or the predaceous bacterium Dello-vibrio. The S layer also helps maintain the shape and envelope rigidity of at least some bacterial cells. It can promote cell adhesion to surface. Pili and fimbriae. Many gram-negative bacteria have short, fine hair-like appendages that are thinner than flagella, are not involved in motility. These are usually called fimbriae. Pilus are similar appendages, about 1 to 10%, that differ from fimbriae in the following ways. Pili often larger than fimbriae. They are genetically determined by sex factor or conjunctive plasmids and are required for bacterial mating. Some bacteria viruses attach specifically to receptor on sex pili at the start of their reproductive cycle. Flagella and motility. Motile bacteria move by use of flagella, thread-like locomotor appendages extending outward from plasma membrane and cell wall. They are central, rigid, about 20 nanometer and up to 15 or 20 meter long. Flagella is hair-like structure. Flagella filaments are made of subunits of a single protein flagellin. The transmission electron microscope studies have shown that flagellum is composed of 3 parts. Filament extend from cell surface to the tip. Basal body embedded in the cell. Hook. Filament is hollow, rigid, cylindrical, made of flagellin. Hook is curved portion slightly wider than filament. Basal body is the most complex part of flagellum. It differs in gram-positive and gram-negative bacteria. The basal body of gram-negative bacteria has 4 rings connected to the central rod, a pair of rings embedded in the cell membrane, and another pair associated with the cell wall. The outer L and P rings associated with the lipopolysaccharide and peptidoglycan respectively. The inner M ring, plasma membrane, and S ring near periplasmic spaces. Gram-positive bacteria have only 2 rings in their basal body. The inner M ring connected to plasma membrane, the outer S ring attached to the peptidoglycan. Since gram-positive bacteria lack the outer pair of rings, it is assumed that only the inner rings are essential for flagellin motion. Bacterial envelope. The number of gram-positive bacteria can form a special resistant dormant structure called endospore. Endospores develop with the vegetative bacteria cells of several genera, Bacillus and Clostridium, Sporosarcina and others. Space formation, sporogenesis or sporulation normally commences when growth ceases due to lack of nutrients. Plasma membrane. Membrane containing both proteins and lipids. Bacteria plasma membrane usually have a rigid proportion of protein than eukaryotic membrane. Lipid from bilayer in members. Bacterial membrane lacks sterols such as cholesterol. Cell membrane are very thin structures, about 5 to 10 nanometer thick. Internal membrane system does not contain membranous organelles like mitochondria or chloroplast. Mesosome are invaginations of plasma membrane in the shape of vesicles, tubules, or lamella. Thus, they may be involved in the cell wall formation during division or play a role in chromosome replication and distribution to daughter cells. Inclusion bodies. A variety of inclusion bodies, granules of organic or inorganic material are present in the cytoplasmic matrix used for storage and also reduced osmotic pressure by tying up molecules in particular form. For example, for polyphosphate granules, cyanophysine granules, and some polyglycogen granules. Example of membrane enclosed inclusion body are polyhydroxybutyrate granules, some glycone, and sulfur granules, carboxymous, and gas vacuoles. Cyanophysine granules are composed of large polypeptide containing approximately equal amount of amino acid, arginine, and aspartic acid. The granules store extra nitrogen for the bacteria. Carboxymous are present in the many cyanobacteria, nitrifying bacteria, and thiobacilli. They are polyhedral, about 100 nanometer in diameter, and containing the enzyme ribulose 1,5-phosphate carboxylase in a paracrystalline arrangement. They serve as a reserve of this enzyme and may be site of carbon dioxide fixation. Gas vacuole. Organic inclusion body present in many cyanobacteria, purple and green photosynthetic bacteria, and a few other aquatic forms such as halobacteria and Theotrix. Gas vacuoles give them buoyancy. Gas vacuoles are aggregate of enormous numbers of small hollow cylindrical structures called gas vesicles. Gas vesicle wall composed of entirely of a single small protein. These protein subunits assemble to form a rigid enclosed cylindrical cylinder that is hollow and impermeable to water but freely permeable to atmospheric gases. Phosphate granules or volatile granules. Volatile granules function as storage reservoir for phosphate, an important component of cell constituents such as nucleic acid. In some cells, they act as energy reserve, and polyphosphate can serve as energy source in reactions. These granules are sometimes called metachromatic granules. Magnetosome is used by some bacteria to orient in the Earth's magnetic field. This inclusion body is containing iron in the form of magnet. Ribosome. Ribosome loosely attached to the plasma membrane, made of both protein and ribonucleic acid. There are cytoprotein synthesis, matrix ribosome synthesis, protein destined to remain within the cell. Plasma membrane ribosome make protein for transport to the outside. Nucleoid. Prokaryotic chromosome is located in an irregularly shaped region called the nucleoid. Composed of about 60% DNA, 30% RNA, and 10% protein by weight. Prokaryote contain a single circle of double-stranded deoxyribonucleic acid, but some have a linear DNA chromosome. Many bacteria possess plasmid in addition to their chromosome. These are double-stranded DNA molecules, usually circular, that can exist and replicate independently of the chromosome or may be integrated with it. In both cases, they are normally inherited or passed to the progeny. Plasmid genes can render bacteria drug resistant, give them new metabolic abilities, make them pathogenic, or endow them with a number of other protein. Reference. Thank you.